crystallographer, and I want to kind of prove to you that we are not a dying breed just yet. So I'm going to talk to you about how I'm using microcrystals, in particular for a protein called GMHA. This protein is from Burkholderia pseudomallei, and the reason I'm interested in it is because this protein also has structural homologs in E. coli and Pseudomonas aeruginosa, really antibiotic-resistant strains, and basically this protein is involved in heptose isomerase, and what it does is that it's a two-step mechanism that does isomerization and then cyclization, and this is really important for the bacterial cell membrane. But what do we already know about it? My supervisor is a GMHA enthusiast, so he was working on this a bit before I was around. But basically what we know already is that there are 4 active sites. Of these, we have 4 zinc ions. Yes, they were present in the structures. And basically, his hypothesis, based on large crystal data, was that it has this really cool negative and positive cooperativity. And this means that when one of the active sites binds a substrate, so S7P, the other one on the opposite side of the protein is then activated. And it does this without any conformational changes. And then also, at the same time, the other binding site in the pair cannot be activated. And how does it do this? They did some mathematical modeling, and they showed it's all connected by water channels, so all of its activation is done through the movement of waters. Obviously, this was all done by modeling and combination of large crystal structures, so the key question is kind of, what's actually going on? We want to actually inhibit this and figure out what's happening. So techniques available for us here. This protein is amazing at crystallizing. That's a novelty in crystallography. And I've been lucky enough in my PhD to use many different microcrystal techniques. And in particular for this protein, I'm focusing mainly on synchrotron serial crystallography. So this is tape drive and foil. But to do this, to get to microcrystals, there's a lot of optimization along the way. So often crystallographers look at something like all down here in this corner, and you kind of think, oh, that's a bit gross. I don't really want to put that under some X-rays. But luckily, this is like gold dust to us microcrystal enthusiasts. So you can optimize this to get to a range of sizes. And for example, for my protein, I've gone through many rounds of optimization, and we can get from something that's relatively large in crystallography terms for microcrystals to something quite small, reproducible, that we can then use for serial crystallography. But what's the advantage of this? Actually, having a small crystal is really, really useful. Often problems with ligand soaking is that you don't get high occupancy in your structures. And when you're doing time-resolved crystallography, you want to make sure you're getting really, really good diffusion of that substrate into the crystal in all of the units in there, so that when you're merging those millions and millions of images, you're going to see a really nice image of what's happening at those time points. So the techniques I'm using are tape drive here, so you can see the crystals, kind of nice long rods come out, and they're fluctuating around in the solution. And what we do is we add ligand, they move down the stream on a tape, and then they're shot with an X-ray. This is a really good technique. It requires about 10 to 40 micron-sized crystals, and we require at least 20,000 crystals to get a data set per time point. For an example on these data sets, just to put it in perspective, I actually took 2.5 million images at every time point. And then the other technique I've used as well is fixed target, in particular foils. These are tiny, tiny, clear solution mylar film, you press your crystals between them, and then basically you go work your way across the grid, taking images. These are both at room temperature. If you want to talk to me about cryo techniques as an alternative, I've also done VMXM and some micro-ED as well. But let's look at some of that foil data. So initially, I added substrate. This was not done with real health and safety in mind. As you can imagine in a beamline at ID29, I was mixing my protein and substrate, sprinting as fast as I could, loading it onto the system, and seeing what happened, basically. This is a very exploratory first method. And you can see when we split the foil, based on like top half of the foil versus bottom half of the foil, we kind of see these differences in density. But most of you will be looking at this going, what on earth is happening? There's lots of random density changes, like all the different active sites are doing different things. We can't really see definitively which one's being activated first versus last. But we can see between the first half of the foil versus the second half of the foil, there are clear changes and clear kind of larger blobs being formed. So we're seeing cyclization happening and product forming in the active sites. This was using a slow mutant of my protein, so I was able to see this much easier. However, obviously, this isn't fantastic data for a PhD. So I then moved on to tape drive. So this is a method by which you get a bit more of a precise estimation of what's happening at each time point. So these are the two active sites that are being shown on the screen. And I did a 500 millisecond time point, 2 seconds, and also 5 seconds. And what we see is actually when we combine all the different data sets is we can actually start to identify intermediates being formed in the protein. So if we look at density coming over on the right side here, we're seeing the first intermediates forming. And in particular, this one up here, we've got this U shape happening, versus down here, we've got this kind of kinking. And this shows two different states of the intermediate. In particular, the zinc up here in that U shape was very close. So this is probably in the process of isomerization, and then immediately it gets kicked off, that zinc. And then we move more towards product. So the product formation here is still seeing some of this density at the top with a ring here. Whereas at the bottom, when we move on to that one, what we'll see is it's been completely detached from those active side chains. So we are starting to see that preferential selection of an active site. So what we're looking at here is actually a completely different mechanism of cooperativity as well. So my supervisor, as I said earlier, predicted that they formed on opposite sides of the protein. These are actually two active sites on the same side of the protein. So now we're getting more evidence as to what's actually happening, and that active site's definitely being selected first. And if we look a bit closer at ligand density, we kind of see that we definitely have some form of intermediates being formed, but as some of you will probably eagle-eyed be aware of, we're kind of getting also merging of states here. So what we're doing at the moment in the process of doing and working with the dials team is trying to do occupancy refinement as well for serial data and make this more of a streamlined process. So this has allowed us to make a new hypothesized cooperativity model where we still believe that, yes, only one active site in a pair can bind the substrate at a time. But, however, we do know now that there is one active site that is preferentially selected. So this one has complete turnover at 5 seconds. There might be a question later about, is that crystal or is it real? Please do come to speak to me at my poster because I also have inhibitors. But also then the second active site is also activated sequentially after. And this one, we see a mixed state at 5 seconds of the intermediate and product being formed. So moving on in the future, I'm looking to try and interrogate what actually happens at those intermediary states. And then if we look at the third site, we actually see this one starting to be activated in the structures because we start to see a phosphate coming through in the density. And obviously, as you can imagine, through refinement and stuff, this is obviously merged images. So the phosphate always gives off a really strong signal. So we know that something's starting to appear. So we can start to see that this one's being activated too. And then this one, meanwhile, I promise there's nothing actually happening there. There's nothing present. It's just waters. So to conclude, we've shown through this work that microcrystals are really useful for understanding protein mechanisms. And previously, isomerization in particular was thought to be too fast a step to capture with this method. However, in crystallo, we do see that everything's happening a little bit slower than in solution, by approximately 20 times. And then also, we're going to be using this GMHA cooperativity mechanism as a focus now for drug development. For the future, we still need to understand the microcrystal techniques better and also make them more accessible to the community because it often takes a lot of workup and you need access to the beamlines in particular that are specialists in this. And also, the final thing is actually understanding the protein crystal dynamics with diffusion and also with ligands. So I'd like to thank all of my supervisory team, and in particular, the TUS group at Southampton, Roberta Steiner and Gerhard for their hard work with RefMac as well, and also Will Stewart and the beamline scientists at ID29 for letting me use it all. Thank you.